Ali, and I will be performing part 1 of the return demonstration, which is paper boat making and specimen receiving and accessioning. But before that, let's put on our PPE. Let's begin making our paper boat by getting a rectangular sheet of glossy paper about the size of 1/8 of a sheet of yellow paper. Proceed to fold it into 3 parts lengthwise, making sure that the folds are well defined. Proceed to fold the paper into 3 parts crosswise. After folding, you now get 9 boxes or divisions on the paper. The next thing to do is fold the corner boxes. When you fold these boxes, allow the perpendicular lines to meet. After doing so, you will now have 4 flaps. After creating these flaps, fold the 2 of them towards each other onto the shorter end of the rectangle. You will now have 2 tabs, and you will then fold these tabs down to stabilize your box. And that is how you make a paper boat. You can now fill this with the embedding medium of your choice. Now let's move on to specimen receiving and accessioning. We begin this process by checking if we have all the necessary forms, namely the histopathology request form, receipt of transaction, and other patient documents. Take note that the histopathology request form must contain the following information: the name of the patient's name, date of birth and sex, medical record number, date and time of collection, type of specimen, the location of the patient, the clinical history of the patient, pre-op and post-op diagnosis, the requesting physician's name, the physician or authorized person's signature on the request form, and the name of address of the physician for referral laboratories. We then proceed to check if the specimen is prefixed in formalin. As you can see, the specimen is indeed prefixed in formalin. After this, we check if the specimen container has the complete information, namely the patient's full name, the medical record number, age and sex of the patient, type of specimen, and the date of time of collection. If the specimen lacks any of these information, then it will be rejected. We then accession the specimen by writing the serial number SP21123, SP for surgical pathology, 21 for the last 2 digits of the year, and 123 is the specimen control number. Moving on, we write all the following details into the logbook: the serial number, the patient's name, age, gender, and room number, the specimen, the name of the pathologist who will be examining the specimen, the date the specimen was received, and the due date. After updating the logbook, we then proceed to fill up the surgical pathology worksheet with the information including the patient's name, age and sex, the sample's accession number, its due date, additional tests if there are any, and the name of the pathologist in charge of examining the tissue. In this step, we will also assist the pathologist during the gross and microscopic examination by writing down his or her findings. During this step, the name of the assisting histotechnologist will be noted down. After performing the gross and microscopic examination, the pathologist's diagnosis will be written down and he or she will affix his or her signature. After completing the accessioning process, we will attach all these files into one folder. This is Ali, and this has been the paper boat making and specimen receiving and accessioning process.
Hi, I'm Danielle and Lamban, and I will be demonstrating to you the different methods of smear preparation. But before that, I will show you the correct way of doing the smear. So, what makes a smear desirable? Number 1, it should have to gradually shift from thick film to thin film. Number 2, it should occupy 2/3 of the glass slide. Number 3, it should be no holes. So, to make a perfect smear, one must follow 4 simple letters: A, S, A, P. A stands for angle, S stands for speed, A stands for amount, and P for pressure. Now let me show you how to do it.
Pour a small amount of the blood on one end of a glass slide and use another glass slide as your spreader with the correct angle and speed, and then you have it. The first method of smear preparation is tricking. Pour a small amount of the sediments on one end of the glass slide, and by using an applicator stick, streak the sediments in a zigzag line throughout the remaining length of the slide. a relatively uniform distribution of the material should be obtained. And then, cover it with a cover slip. After that, label your slide. Type of specimen, current year, and the specimen control number. And then, you have it. The next method is spreading. Transfer a small amount of sediment on one end of the glass slide. And by using an applicator stick, spread the sediments throughout the slide, similar to how butter is spread on the bread. And then, label your slide. And there you have it. Next method is pull apart. Place a drop of sediments at the one end of the glass slide. And then, cover it with the end of another glass slide. Allow the sediments to spread evenly between the attached surfaces of the two slides. With a single uninterrupted motion, pull the two slides apart in an opposite direction. And then, cover the smear with a cover slip. And don't forget to label your slide. And there you have it. The last method that we have is the touch or impression smear. Allow the freshly cut surface to come in contact with the surface of the glass slide. Apply gentle pressure, allowing the cells to be transferred directly to the cell. And then, cover the smear with a cover slip. And don't forget to label your slide with the type of specimen, current year, and the specimen control number. And there you have it.
Those are the different methods of smear preparation.
I am Rouella Maglana and I will be performing the proper fixation of tissue samples. Let's go! So here's our sample at approximately 50 grams, a pinkish color and a smooth texture. That is 11 by 7 centimeters with a slightly slimy consistency. And now we are going to cut a representative specimen into 2 cm squared but not more than 4 mm in thickness. And that is in order for the fixative to penetrate the sample thoroughly. Fixation is the preservation of biological tissues from decay due to autolysis or putrefaction. It terminates any ongoing biochemical reactions and may also increase the treated tissue's mechanical strength and stability. Since I don't have a tissue cassette, I will be putting the specimen in a gauze pad instead. I have also opted to label the gauze pad with my code Marites. Yeah, you can call me Tessie. I will now put the representative specimen onto the gauze pad using my tweezers for sanitary purposes. Oops! Again, since I don't have tissue cassettes, I opted to use gauze pads instead. But in a laboratory setting, tissue cassettes are totally resistant to the chemical action of histological solvents. Now, using a 500 ml beaker, we will now fill the beaker with 10% formalin up to the 400 ml mark. Fixative ratio of this specimen should be 1:10 since it is a small specimen. As you can see, I have already tied the gauze pad to secure the specimen inside. The specimen must be fixated within 20 to 30 minutes after cutting off blood supply and 6 to 48 hours fixation, overnight for routine processing, and for large organs at least a week. Room temperature should be at least 45 degrees Celsius or up to 65 degrees Celsius in a microwave. Increased temperature will speed up the fixation rate. However, the presence of heat will also speed up the autolysis. Therefore, as much as possible, we limit our temperature around 18 to 45 degrees Celsius. And there you have it, the proper fixation of tissue samples. Thank you for going through this with me. Till the next time!
Hi, I'm Jodi, and I will be doing an optional process in the routine procedure in the Histopathology Laboratory, which is decalcification. Here are the materials: the specimen, beaker, the calcifying agent, and other materials needed for the manual procedure. First is to make a container for the specimen or the tissue that is needed to be decalcified. After which, we will get the specimen or the tissue needed to be decalcified and transfer it to the prepared gauze. When wrapping the tissue or the specimen, it should be the gauze is loosely wrapped around it. To secure it, you need to tie it with a string. When you are done securing it, this time you need to tie the other side of the string to the rod so it will look like hanging, and you need to also to make sure the length that the specimen will not touch the bottom of the beaker. Here is what it should look like. So again, it will not touch at the bottom of the beaker. Next step is to use the decalcifying agent, so we need to transfer it onto the beaker. Please take note that the ratio for the decalcifying agent to the tissue or specimen is 20:1. Proceeding, we will now submerge the wrapped specimen or tissue to the decalcifying agent. Please take note that you will need to wait 12 to 24 hours for it to be decalcified. Therefore, please make sure to store it properly. So that's it for the decalcification process. Thank you so much.
Hi, my name is Ronamae Del Mar and I'm in charge for the dehydration process. And now that I'm done wearing my PPE, let's get started! So before we actually proceed to the dehydration procedure, let me first give you an overview of the materials to be used and some additional notes that are very vital in this procedure. Let's proceed with the materials. We need the specimen, of course, the decalcifying agent, which is specifically 70%, 90%, and 100% ethanol. So as for the 100% ethanol, we need 4 sets of this ethanol since we are going to have 4 changes. Now, we also need the forceps, and since we don't have the forceps, I'm gonna improvise on the chopsticks. We need a beaker, and since we don't have that at home, I'm going to improvise on the glass. For the tissue cassettes, I'm going to improvise it as a gauze pad with string, and we need labels to know what percentage of the calcifying agent is being used, and of course, very important, your PPEs. More importantly, to ensure that you are procuring the best results, you would need to take note of the following. The fluid to tissue ratio should be 10:1, the ideal temperature should be 37 degrees Celsius, and the specimen should not be more than 4 mm thick. So let us assume that this tissue has already been processed under the previous step, and this is what I received and is now ready for the dehydration step. Also, the tissue was wrapped in a gauze pad and tied by a thread since there is no tissue cassettes available. So for the first step of dehydration, we are going to submerge our sample in a lower concentration of dehydrating agent, and in this case, 70% ethanol. So we submerge our sample in 70% ethanol for 15 minutes. Now step 2, after 15 minutes, we submerge our sample in a higher concentration, which is the 90% ethanol. Oh, and since I don't have any forceps at home, to improvise, I use chopsticks. Okay, so this tissue is being submerged, and it's being submerged for 15 minutes in 90% ethanol. So for our third step, we are going to submerge it in our first change of absolute ethanol. So we're going to submerge it in 100% ethanol for another 15 minutes. So after waiting for 15 minutes, let us now proceed to step 4, which is our second change of absolute ethanol. So we're going to submerge it in the second change of absolute or 100% ethanol for another 15 minutes. So after waiting 15 minutes, we now proceed to our step 5. So our step 5 is our third change of the 100% ethanol or our absolute ethanol. So now we submerge this tissue in this third change for another 30 minutes. So after waiting 30 minutes, we are now going to proceed to our last and sixth step, which is the fourth 100% ethanol change. Now we're going to change it into 100% ethanol again, and we submerge it this time for 45 minutes. After 45 minutes of waiting, we can now say our specimen has been dehydrated. And since we follow the ideal time, ideal fluid to tissue ratio of 10:1, and the ideal temperature, we can now say that our sample is ready to go to the next step. But why do we need to fully dehydrate the tissue, you ask? As a medtech, it is very important that you should know why you are doing dehydration and its mechanisms behind it. Well, from the word dehydration itself, it simply means to remove the water from the tissue specimen. Now, this water is removed to avoid further changes and other complications that may arise in the following steps. Not only may it arise complications in other steps, it may also lead to the degradation of the quality of our results. This is Rona, and this has been your dehydration process. Thank you!
Hi, I'm Hannah Eng Fo, and I'll be performing the clearing technique. Before anything else, we have to make sure we are wearing our complete PPE. Let's go! So these are the materials that I will be using, and these are the reagents. However, this is not real saline. So let us assume that this tissue has already gone through all the tissue processes up until dehydration, and now it is ready for clearing. So place your tissue specimen into a gauze pad and wrap that around and tie it securely with a thick string. So I will be speeding up this process for time's sake. After tying it securely, this tissue specimen will then be attached into a rod. For this case, I am using a chopstick. You will tie that, and now this tissue is ready for immersion. For your first immersion, your tissue specimen will sit in saline for 20 minutes. You have to make sure that your reagent is 10 times the volume of the specimen. You have to make sure that your tissue specimen is completely immersed into the reagent. After waiting for 20 minutes, you will be taking out your tissue specimen and placing it into another set of saline. And again, you will be waiting for 20 minutes. After waiting for 20 minutes, you will be taking it out again and placing it into another set of saline, but this time you will be immersing it for 45 minutes. After waiting for 45 minutes, we will be taking out our specimen from the reagent. At this point, the tissue has already completed the clearing process. So we will untie the string and open up the gauze to check and see the appearance of the tissue. We are expecting to see a translucent or transparent or clear tissue appearance. As you can see, the tissue specimen has successfully completed the clearing process, and at this point we will be taking it out and placing it into a tissue paper to drain it or dry it from any excess reagent that is sitting on the surface of the tissue. After which, we will prepare the tissue specimen for storage. So we will take it out of the paper towel and place it inside a tin foil as of this moment to store it for the next histologic procedure. Always remember to label your specimen and observe proper labeling. That would be all. Thank you so much. See you in our next video.